Biosynthesis of fatty acid, part 3rd. In this third lecture of biosynthesis of fatty acid, we are going to study about the priming phase and elongation phase of fatty acyl chain. Let us start. Priming of fatty acid synthesis by acetyl coenzyme A. The two SH group present on the fatty acid enzyme complex must be charged with the correct acyl group, and this charging is known as the priming of the system, and it takes place in two steps. First step of the priming phase. In the first step, the acetyl group of the acetyl coenzyme A is transferred to the SH group of the beta ketoacyl ACP synthase, and this reaction is catalyzed by acetyl coenzyme ATP transacetylase. Second step of the priming phase. In the second step of the priming phase, the malonyl group from malonyl coenzyme is transferred to the SH group of the acyl carrier protein ACP, and this reaction is catalyzed by the enzyme malonyl coenzyme A ACP transferase MT. Elongation phase, first round. In the charged synthase complex, the acetyl and the malonyl groups are very close to each other and are activated for the chain lengthening process. And this process, this elongation phase, consists of following four steps. First step is the condensation. In this first step of the formation of fatty acid chain, the activated acetyl group and the activated malonyl group condense to form the acetoacetyl group, and this acetoacetyl group bound to ACP through phosphopentethene SH group. Elongation phase, first round. In this elongation phase, first round, second step is the reduction of the carbonyl group. The acetoacetyl group formed in the condensation step undergoes reduction of the carbonyl group at C3 to form beta hydroxybutyryl ACP. This reaction is catalyzed by the enzyme beta ketoacyl ACP reductase, and the electron donor is NADPH. The NADPH is the reducing agent, where NAD plus is the oxidizing agent in beta oxidation. In the third step of the elongation phase, which is the dehydration step, one molecule of water are removed from second carbon and third carbon of the beta hydroxybutyryl ACP to yields a double bond in the product trans delta butynyl ACP, also known as crotonyl ACP. The enzyme that catalyzes this dehydration reaction is beta hydroxyacyl ACP dehydratase. In the fourth step of the elongation phase, which is the reduction of the double bond, in this step, the double bond of the trans delta butynyl ACP is reduced to form butyryl ACP, and this reaction is catalyzed by the enzyme enoyl ACP reductase. Again, NADPH is the electron donor or the reductant. These four reactions, condensation, First reduction, dehydration, and then again reduction. These four reactions together complete the first round of the elongation cycle. Thus, after the first round of elongation, 4 carbon butyryl precursor of palmitate has been synthesized from the 2 carbon acetyl and 3 carbon malonyl unit. Successive round. During the second round of elongation, the butyryl group is now transferred from this phosphopentethene SH group of ACP to the cysteine SH group of the beta keto acyl ACP synthase to start the next cycle of reaction that lengthens the chain by 2 more carbon. Another malonyl group is linked to the now vacant position of the phosphopentethene SH group of the ACP. Now condensation occur at the butyryl group which exists at the condensation occur at the butyryl group existing at the as earlier the acetyl group in the first round is linked to the 2 carbon of malonyl ACP with the release of one molecule of carbon dioxide. And this condensation of the butyryl group with the malonyl coenzyme forms the 6 carbon beta keto acyl ACP. The 6 carbon fatty acyl ACP is now ready for the third round of elongation. 7 such cycle of elongation occur, and as a result of these 7 successive cycles of elongation, 16 carbon palmitate gets produced, and for this reaction 8 acetyl coenzyme is required. 7 molecule of ATP and 14 molecule of NADPH is required for the formation of 1 molecule of palmitate. The ATP is required to attach carbon dioxide to acetyl coenzyme A to produce the malonyl coenzyme A, while the NADPH is required to reduce the double bond to form the corresponding saturated fatty acyl group. For further reading, I would like to suggest Books of Biochemistry by S.N. Gupta and Fundamental of Biochemistry by S. Chan. Jain and Jain, S. Chan publication book. Thank you for watching.